Okay, I think we can start. Still some people joining, but it's almost not growing too much now. So welcome to this second part of the data track session. We have for this second period, we have five contributions, 5 presentations. Probably you were here the first session. That was very interesting. I think that this is going to be as interesting as the previous one. And as always, we'll be really tight on time, even though that session is a bit longer than the first one. So without much delay, I will present the first one. I just remind before that if I don't mess it up, I will be asking the questions in your behalf, but you have this live Q&A chat to put your questions, and at the end I will try to translate or to read with some significance. Okay, as I said, we will start. First speaker in this second session is Wesley Qian or Qian from the University of Illinois in the USA, and I think it's correct, yes. And he's going to talk about batch equalization with generative adversarial network. Wesley, the floor is yours. Wesley?
Cool. Yeah, should I share my screen or?
Are you sharing? Okay.
Okay. One sec. Right. Thank you, everyone. So today I will talk a little bit about batch equalization with generative adversarial network. I'm a PhD student in University of Illinois, and this work is actually done partially when I was interning at Google. So this is a collaboration with some of the folks at Google Research, as well as some folks from the Imperial College, which we look at some of their very interesting data for malaria parasites. So to motivate the problem a little bit, today we have this high throughput technology that allow us to sort of do lots and lots of experiments each week. And so for each week, scientists can do, say, 15 or 60 compounds or different perturbations of the compounds, and then image them. And then the second week, they can do another batch of different perturbations. And the third week, they can do a lot more other perturbations. But so essentially, in each week, they can do a set of different perturbations. But this actually, because you are not able to do all the perturbation in one batch, that gives you this problem of batching effect. So if we look at this BCO21 data set, and if we just, so each row is the data collected for 1 week. And then if we look at the color profiles, like, so for week 2, this color is definitely very different. And then for later weeks, we see sort of this more subtle difference where some of the batches are dimmer. So essentially, when you're sort of doing downstream analysis, this kind of batch effects would sort of correlate or entangle with the sort of biological features that you want to sort of recover. So this would give you trouble. So if we, so one experiment that we sort of did, or teammates of mine did, is we look at the sort of an SLA data, which we have the image embeddings, and then we look at the 2D visualization of the embeddings. So if we look at a clustering of the disease state or healthy state, we see this nice little clustering.
Excuse me, Wesley. I'm not sure if we are being the right.
Oh, okay. You're not.
We are viewing an Ayöz screen, not yours. I don't know which one.
Let me try again.
I think you can control the one that.
Oh, okay. Perfect. Okay, yes, that's perfect.
That's it? Okay.
Yes. Yes. So okay, so let me go back to this one then. So because we have this batch effect, so the batch effect, if we put all the data together, what we see is that, so each row is 1 week of the data. And then we see that week 1 is definitely a little bit bluer than the others. And then if we look at the later weeks of the rows, we see that some of the green is dimmer than the others. So this kind of batch effects would give us troubles if we go to downstream analysis where sort of the biological perturbation is actually entangled with the technical issue caused by or the technical artifacts caused by the batch effects. So in here, what we are showing is that we look at the embeddings of the image embeddings, and we do a testing, project it back to a lower dimensions and visualize it in 2D. And then what we see is that we see this little clusters of different disease states or healthy state. But if we actually look at but for the same state, some of them are not clustered together. And if we actually look at sort of relabel this into the batch, what we see is that we actually see a larger cluster driven by the batch membership instead of the sort of the more healthy disease, instead of the biological state of the cell. So essentially what we want is we want to sort of correct for each batch effect so that the cluster is not sort of the batch effect or the batch membership, the variance is the variance in the embedding is encoding less batch information, but more sort of the biological information that we care about. And so the way we sort of think about this is that there is actually a lot of literature going into batch normalization. What it does is essentially we try to project all the data from, so if we have a different batch of data sort of doing different experiment conditions, we try to project it into a sort of a neutral state or baseline state, and then we do an analysis there. So that's what we usually call batch normalization. And in this work, what we try to do is we try to do something called batch equalization. So instead of projecting everything down to a neutral state or baseline state, we try to actually be able to transfer between batches. So if we're actually we are able to transfer batch 2 to batch 1, batch 3 to batch 1, and essentially convert all the batches into the same batch, then we can actually just analyze them together once we can equalize them. And the way we are able to do this is called style transfer. So we essentially formulate this as a style transfer. So in the deep learning literature, there's this framework called StarGAN, where essentially you can sort of you input an image of a star space, and then you can actually change their hair colors into different hair colors, and then even change their facial expression. So this is very powerful because essentially you can do this kind of style transfer given an image, and then you keep the semantic contents of the image but change the style. And in here, what we can do is essentially formulate each batch as a style, and then so essentially we can sort of think about we input a cell image from any of the batch, and then we say transfer it back to batch 1, batch 2, batch 3, and batch 4. So this allows us to sort of have a framework to sort of transfer between treating each style batch as a style, each batch as a style. This allows us to do style transfer. And sort of you can look into the preceding papers a little bit more, but sort of to give a very high level architecture. So in the StarGAN, we have essentially 3 laws in the original StarGAN. So one is what we call a reversal laws, which essentially give a realistic look to the image, such that we're able to sort of generate image that looks very real. And then the second one is what we call domain classification loss. So this is sort of making sure that the network is, the generative model is actually converting the image to the right target style or the right batch member. And then the third one is the reconstruction loss. And this is quite important because this essentially allows you to keep the semantic content of the image such that it requires the generated image can be converted back to the original image, so that you cannot the model can also just provide a trivial solution. It has to keep the semantic content such that it is able to convert back to the original image. So this is the original StarGAN, and it's actually very powerful. So if we actually just directly use this framework to apply on our data, so we have an image from batch 0 and we try to transfer it back to different batches, and we see that the sort of transfer the green one to a blue one, the blue batches that we just saw. And then we also see that it actually is, the image looks very, very real, and there's a lot of very little artifacts in here. However, if we look at the difference between sort of what are the model making the modification, what we saw is that for batch 7, because it actually has a very high confluency in the experiments, we see a lot of hallucinating cells in here. And then we also see that sort of the cell size is being shrinked because the batch A and batch I just have a smaller cell area. So essentially the StarGAN is a very powerful tool that sort of are able to capture the statistical meaning as the statistical patterns in the data. But this is maybe not the kind of things that biologists would want to see, like what does the hallucinating cells mean? So why are you changing the cell area? Because this could really mean something based on the perturbation of the experiment you did. And so what we sort of, so based on that, we added two losses to look at this. So the first one is the feature disentanglement loss. So essentially, if we look at the one thing I forgot to mention is that we're actually doing all the training on the negative control, and even on the negative control, we still see this biological feature that are entangled with the batch membership. So essentially, what we want to do is we want to have a way to only transfer the batch membership, but not the biological features. So what we have is something called a feature disentanglement loss. So what this means is that when we look at the generated image, we also we can actually control some set of features that is not being, we request or we ask the generator to not change this kind of feature, like the cell size or the number of cells in the data, in the image. So this allows us to sort of have a way to explicitly state that this feature should not be changes based on the batch changes. So this is the first feature add-on that we did. And the second one, what we did is what we call local variation loss. So this is actually based on a very simple observation on the on the batch effects. A lot of the technical batch effects that we saw is actually uniform because it's like a variation in the illumination or the staining intensity. So all this thing is generally pretty uniform. So what we do here is that instead of allowing the generator to modify the image with a lot of variance, we actually we allow the generator to make any sort of high or low modification as they want, but sort of the modification at each pixel need to be sort of the variation need to be small. So this is the second sort of constraint we impose to the models. And if we sort of look at what we got after we so this is what we saw before, which is with the original StarGAN, which we see this sort of hallucinating cell and shrinking cell size. Once we sort of incorporating this sort of biological driven tricks into these models, we see that sort of this hallucinating cells goes away, but in the same time, we still keep this sort of contextual or nonlinear changes in the image, which is really nice. This is really promising results. So we look at another sort of data set that we are sort of working on, which is the malaria parasite images. So in here, what we are sort of doing is that in this experiments data set, we actually we've done this over a span of half a year almost. And then we actually see a lot of batch effects in here. So the first row is the original image collected from each date. And then we are trying to sort of equal, and then you can see that some of them are darker and some of them are very bright. And then so what we do here is we try to convert all the images to the last date. So this column here. So what we see is that once the model is actually able to sort of also work on bright field images, and then a different data set, we sort of convert everything into a sort of a similar scale of gray here. So this is and we without even tricking any of the hyperparameters, because this is a very complicated model. So we were a little bit worried about hyperparameter trimming, but without sort of a lot of hyperparameter trimming, we can actually just directly apply the same model sort of training procedures to this new data set, and it just works. So this is pretty exciting. And based on this, we sort of want to do a little bit about the quantitative analysis here. So what we did is we looked at how predictive is the embeddings how much we can predict the batch membership of the embeddings. So if we are able to sort of equalize the batch, we are expected to have less predictive power here. So we see that sort of by converting to different batches, we see a smaller accuracy or lower accuracy comparing to the original image embeddings. And if we look at the visualization of the 2D testing embeddings, what we saw is that sort of in the original embeddings, we see this cluster of the batch one here, but once we do the equalization, we see the sort of the 2D visualization is much more spread out in the bottom here. And then the second one,
Wesley, excuse me, one more minute, please.
Okay, sounds good. So the last question is that if we are able to sort of correct for the batch request, are we actually changing the biology? So what we see here is that when we go back to the original biological task, we are still able to solve the biological task, which means that the biological information is not lost. And because we're able to sort of decluster the data here, we are able to, when we use this 1 year neighbor to do the MOA prediction, we're actually able to predict better, have a better performance here. Sort of just to wrap up a little bit in here, we use this batch equalization strategy to do the batch normalization, and then we formulate this as a style transfer problem, and then we use the star gain with some biological driven tricks, and we're able to correct for the batch variation in the embeddings, and we're able to improve the phenotypic prediction, and so the biological feature is not lost. And so there's a lot of application of this like outlier detection, but this is I think this is a very interesting framework for us to do the impossible experiments, to think about, can we actually do the same experiment in the same day? So, or I think about more crazy ideas, like I wonder if I apply drug A to this cell and it looks like this. What if I apply drug B to this cell? What would be the difference? So this sort of style transfer or this framework allow us to sort of think about this sort of hypothesis questions, and it allows us to do this hypothesis generation. So that's everything. Thanks for your time.
Thank you, Wesley. It's a bit late, and actually there is, as far as I see, there is no questions in the chat. Just correct me if this is not correct, because I had problems in the previous session. I have a quick one. I understand that you don't need any relationship between the batches, so they could be just different samples of the same thing, or you need some kind of control spot somewhere to relate these things.
Yeah. So there's no. So we do sort of the one thing is that we require is we need to have a negative control for each batch, such that sort of, because you don't want to have, if you don't have the negative control, then the batch transformation would be capturing the sort of the drug effect instead of the batch effect. So that's, I think, the only requirements that we need, and then sort of, and you don't really need this pair relationship in any way. So that's sort of the powerfulness of this framework.
So you need something that has no biological information just to check. That's the point.
Yeah.
Okay, I got a question from Grigory Sukurov that say, thanks for the nice talk. Could you please elaborate how your GAN projects batch 0 to batch 1? What is the input of the GAN?
Oh yeah. So what we do is we have, the input to the GAN is an image from batch 0. And then plus the targeted batch. So essentially the function would be the input would be the image and a number indicating which batch we are transferring to. So that's and then we essentially pad that number around the image, and then we feed that into the neural network.
Okay, I don't see much more questions. So I probably should go on. We're on time with the next talk. I'm not sure if Elliot is there.
I'm here.
Oh, yes, you're there. Thank you. So next speaker is Elliot Layne from McGill University in Montreal, that is going to talk about, as the first talk on phylogeny today, supervised learning on phylogenetical distributed data. Elliot, the floor is yours.
Perfect, thank you. I'll just flip back to the first slide if that's all right. Mm,
You should be able to control it.
yeah. There we go. Perfect. Thank you. All right. Hello, everyone. So yes, my name is Elliot Layne. I'm a doctoral student at Mathieu Blanchet's lab at McGill University. And today I'm just going to be discussing some of our recent work on supervised learning with phylogenetically distributed data. So the core problem we were considering here was how to best go about making phenotype predictions for a set of organisms by use of machine learning models when the input data set was a set of genomes, each of which coming from a different species of origin. Now, usually when we're doing supervised machine learning, of course, we're working with the IID assumption, that is that our data is independent and identically distributed. But biology is messy, and when our data set is a set of different species, this isn't actually the case in reality. These species will share phylogenetic relationships. So an example, we just have a small phylogenetic tree pictured here of 6 fungus species. And we might imagine, for example, in the case of an imbalanced tree, that a predictor that is trained on a data set mostly coming from ascomycetes might make poor predictions on fungi from basidiomycetes. And it seems plausible that the ideal function relating genotype to phenotype could actually be different in different parts of this tree. So to formalize this just a little bit, we have just a 3 species phylogenetic tree here, human, mouse, and rat. In our typical supervised learning setting, we would be training one predictor, right? That's going to try and find the ideal parameters to make predictions on all of human, mouse, and rat. And we imagine if this relationship between genotype and phenotype is changing in different parts of the tree, that it might actually make more sense to train a family of closely related but different predictors. In particular, one for each node in the tree. So at node D here, our internal node, we'd be wanting to find the predictor f of D, which is the ideal predictor for the species mouse and rat. At node C, we'd be wanting f of C to just make ideal predictions on rat. So there are a bunch of ways that people normally deal with this, because this task of phenotype prediction is one that's really well studied. A common one, which often works pretty well, is to simply ignore the fact that there are phylogenetic relationships, which is to just do standard supervised machine learning. Another common one would be to split our data set into multiple sub-data sets. This can often work really well. Some of the drawbacks here, of course, that each data set will then be smaller, which could impact the top model performance. And it's not always obvious how and when we should divide data sets into sub-data sets. And then finally, there's a whole collection of methods that involve some sort of encoding the phylogenetic placement and then adding them to the training features to try and account for any differences that arise from phylogeny. So linear mixed models would be an example of approaches in this category. So our core contribution was what we're calling the dendronet method, which is a way of training this family of related predictors all we imagine that predictors, evolution is a gradual process. So even if we are going to have a family of predictors instead of a single one, they should be pretty close together. And in particular, if we look at our example phylogenetic tree here, we imagine that the predictor for mouse and rat should probably be quite similar and possibly more similar than the predictor to human. So to enforce this, we just essentially add another term to our loss function. So we have a 2-term loss function when we're training this. The first one is what we call the prediction loss, which is just the sum of all the errors made on predictions, whether that be a cross entropy or a mean squared error, whatever suits the problem you're working with. And our second term is what we call the dendronet loss. So this is what's ensuring that our family of predictors are closely related and that ones that are closer together in the phylogenetic tree should be more similar than those that are far apart. And this is just the sum in the difference of the parameters for our predictors at each node that share an edge, so a parent-child node. And then we scale this by some hyperparameter lambda, which needs to be tuned, which we'll talk a little bit more about later. So to give an example of how a dendronet architecture would look, this would match our 3 species tree, human, mouse, and rat. For our F here, our family of predictors, we're just using a simple fully connected neural network architecture. You can see at the root, we instantiate this weight vector W. So this is just a normal trainable weight vector that you would use to parameterize a fully connected neural network. And then along each edge in our phylogenetic tree, we instantiate a delta vector. So these delta vectors have the same size and shape as W. They're also trainable. And at each leaf, how we make predictions is we pass as input X at the current node, which represents some genome or genetic input. And then our neural network is parameterized by W plus the sum of all delta vectors along the path from the root to the leaf. So to make a prediction at C, which had our species rat, our effective weights are W plus delta D plus delta C. And you can see what our two-term loss function looks like here, where we just have the mean squared error of every prediction of some phenotype Y, as well as the L1 norm of all of our delta vectors. So to examine dendronet's performance, we performed 3 experiments. I'm going to talk about 2 of them here today. For the first one, we just really wanted to confirm, like, is it possible to actually model an evolving relationship between genotype and phenotype at all? And in particular, is it possible to do this without overfitting when we have parameters that are really only paying attention to small subtrees? So we just generated a simulated phylogenetic tree. We instantiated a parameter vector A at the root, and we allowed it to evolve along the edges in our phylogenetic tree. And then at each leaf, we generated a feature vector X and used both the independent feature vectors X and these phylogenetically related parameter vector A to produce our Y values. And we trained a fully connected neural network, as well as a dendronet family of fully connected neural networks, and just took a look at what happened. So we ran a bunch of experiments with different lambda values, where lambda scales up or down the penalty given to dendronet families when they have differences in their parameter values. So like how much the model is allowed to evolve, for lack of a better word. And then we also varied the mutation rate, which actually affected the size of the updates on our A, which generated the values we were trying to predict. So with low mutation, we can see that our baseline model performed as well as any dendronet model, which makes sense. We added roughly 0.8 average amount of noise to the label. So that's why everything caps out at 0.8 mean squared error. And as the mutation increased, we can see that dendronet here with a dotted line at the top where we had no regularization overfit the data at all mutation levels. This makes sense. We should require some amount of regularization to prevent the model from overfitting. The highly regularized dendronet and the baseline model started to perform very poorly. Once the mutation rate increased, they weren't able to model these changing parameters. But our nicely tuned dendronet, the Goldilocks values for the hyperparameter lambda, still modeled the evolving A parameters quite well. On the right panel, we just have a little peek at what the loss looked like throughout training. At the beginning, there was no dendronet loss, as our family of models all shared the exact same values for their parameters, but there's high training loss. And over the first 1,000 epochs, our training loss comes down pretty drastically. Our dendronet loss increases as the model finds the spots where it needs to mutate its parameters to fit the evolving A values, and then everything kind of plateaus off eventually. The second experiment I'm going to talk about was on a real-world data set comprised of a bunch of fungus species. And we tried to relate genetic features of these fungus species to predict their trophic level. And in this instance, trophic level was the type of substrate that they were able to feed on. Like, for example, whether it was a fungus that fed on dead host material or living host material. These were all plant pathogens, so relevant to the plants that they feed on. For features, we used counts of different secondary metabolite genes, which is a gene family that relates to the ability of fungus to break down cell walls. So there was some collaborators at UBC kind of pointed us towards this as being a relevant feature set for this task. And we just compared dendronet, this time using a logistic regression architecture as its base F model for the family of functions, with what we considered to be the most relevant comparison set, including the same logistic regression architecture without having a family of them instantiated, linear mixed models, and other kind of similar approaches that typically deal with hierarchically structured data, as well as a parsimony approach, which we threw in because we wanted to capture to what extent dendronet was just modeling phylogenetic signal instead of actual changes in the feature signal. And then when we looked at the results, we saw that across the 4 trophic levels, dendronet, which is in red here, I apologize for the small font, overall dendronet was the best method, and in particular on obligatory biotrophs and necrotrophs, dendronet had significant increases in AUC on the test set. There was also an antibiotic resistance experiment we did relating bacterial genetic data, antibiotic resistance. We saw positive results there. I didn't have time to put it in this presentation, but it's included in the paper. So if you're interested, I'd highly recommend that you check it out. So just to quickly sum up, dendronet is a statistically principled way of accounting for non-IID phylogenetic structures inside of biological data. One of the big pluses to our approach that we really liked was that you can use any kind of model architecture as the submodel that you instantiate a family of. So for our fungus experiment, we just used a logistic regressor, which ends up being fairly similar in approach to linear mixed model. But as you saw with our simulated data set experiment, you can throw in a neural network, a convolutional neural net, really anything can be trained in this way. It gets a little more computationally intensive as you scale up the size of the subnetwork, obviously. We saw increased model performance on our simulated data set, as well as 2 real world experiments, the fungi and the antibiotic resistance. The key kind of drawback to this method is that, of course, the actual number of trainable parameters scales up linearly with the size of your data set. So there will be really massive biological data sets where this can become sort of a bottleneck issue. And then in terms of what we want to try and accomplish next with this, we'd like to expand this kind of tree-based regularization beyond phylogenetic networks to data that is related perhaps by more complicated hierarchies, including like non-tree directed acyclic graph structures. So thank you everyone for coming to listen today. I want to thank my colleagues at the McGill Lab and our collaborators at UBC. And then we had some funding from the BioSafe project, so thanks to them as well. And I'm happy to take any questions now.
Try to start my video anyway. Thank you, Elliot. I'm sorry I was muted. We have a first question by Aljaman Dik. I don't know if I can pronounce that. The question is cool about the comment. Cool approach. You deal with the structure in the phylogeny. Another aspect of phylogeny which will impact supervised machine learning will be biased in terms of how your cases are sampled from the phylogeny. Would your approach correct for that? And would it be possible to extend it in that direction?
So I guess just trying to interpret that question, I'm going to assume that biases would be in terms of where we're sampling from, like perhaps sampling from one portion of the tree more than others. So I think the approach doesn't explicitly correct for that. It was one of the motivating issues we had, was that if there are differences in how parts of the trees should be handled, we expect that smaller parts would be essentially just modeled more poorly. So with the correct amount of regularization, like if it's low enough, essentially, the approach can figure out that arbitrarily small subtrees should be given different treatment than the overall tree. It's not kind of explicitly clear how to set the regularization to balance between modeling evolution correctly, whereas also like a tree may be smaller than it is in reality. So the contribution to the prediction loss could be smaller than it really should be. So it's capable of fitting it, but there'd perhaps be more work to do to account for how to balance the increase in dendronet loss versus the decrease in prediction loss when a subtree looks smaller than it really is, if that makes sense.
There's another comment by Alt, is that she said indeed sampling more from certain parts of the tree. Is that what you were saying? I think.
Yeah.
Okay. So we are on time, so I'll probably finish here the questions and pass over to the next talk. Thank you, Elliot. Thank you very much for the talk. Next talk is also in the world of phylogeny, if I am not wrong. So this is by Jose Barba-Montoya from Temple University in Philadelphia, and the talk is about using a GTR plus gamma substitution model for dating sequence divergences when stationary and time reversibility assumptions are violated. So Jose, go ahead, the floor is yours. You're muted.
Okay, thank you. Hello, everybody. Thanks for attending the talk, which is entitled Using a GTR plus gamma substitution model for dating sequence divergence when stationarity and time reversibility assumptions are violated. This is a recent study we developed in the lab of Professor Kumar at Temple in collaboration with Qian Tao. Okay, as we know, the substitution of amino acids and nucleotides is very complex and it varies among loci among lineages and also over time. Also, so for that, there have been a lot of effort on trying to develop methods that can account for this heterogeneity of the substitution pattern. And as we know, the data sets are growing in number of loci and gene and lineages, so that increases the complexity of the substitution process on the data we are currently analyzing. So here we're going to focus on the GTR substitution model, which provides for different rates and transition and transversional substitutions, and also in equal base frequencies. As you can see, we can see the model, and it has 9 parameters. And this model, the GTR model, assumes that the instantaneous rate of change between base I and base J is equal to base J from base J to I. This is called the time reversibility assumption. As you can see, this assumption is not real, biologically real, but is used for mathematical convenience. Also, this model assumes a stationarity of the substitution process. That means that the substitution process, the assumption doesn't change along the tree. So it assumes that, for example, the composition, GC composition, and the substitution process is the same along the whole tree. And as we know also in real data, that is not the case. So in general, the trees are as the tree on the right, which show different base compositions along the tree. So we wanted also, as we know this complexity is well appreciated, but as we know, most of the studies for molecular dating employ the GTR class of substitution models. From a survey of 141 studies published from 2015 to 2017 showed that 77% of the studies use the GTR substitution model plus gamma plus I, which gamma assumes is used to prior a distribution of the different substitution rates, and I is the number of the proportion of invariant sites. These two parameters can be added to any other model. As we can see, most of them use the GTR, which have 9 parameters, but in general, only 1% use Kimura 880, which has a very low number of parameters. But in general, all of these assume stationarity and time reversibility of the substitution model. So the main question of this study was because in a previous study of the bias cause of the GTR substitution class on divergence time estimation, they show that the bias of using this model or complex or simple models has a limited impact on divergence time estimates. So we wanted to see if this trend holds when we consider the GTR substitution model when the stationarity assumption and time reversibility are violated. For that, we quantify the bias of using the GTR plus gamma model, and we analyze simulated data that were evolved on their without the reversibility and the stationarity process. Okay, for that we simulated the data. We simulated 300 nucleotide alignments that were in which the substitution process was GTR, non-stationary, and non-reversible. For that, we simulated 50 phylogenies under the autocorrelated model and another 50 on the independent rate models. And for each phylogeny, we simulated a GTR alignment, a non-reversible, and a non-stationary. So in total, we have 150 autocorrelated rate simulated data and 150 independent rate simulated data. And this is like the explanation of the 3 simulated data. So as we show the non-stationary, we simulated 2 clades that had very high and the other one very low substitution rate in comparison with the whole tree, and we wanted to see if this will have an effect. And for that, we use relaxed cloud methods, which is the Bayesian, MCMC3, and real-time, relative times. So the results we found, interestingly, we found that here on the right, we are showing the comparison of times using the GTR model for the GTR data against the non-reversible data using the GTR data. And we wanted to see how big was the bias when we are using a wrong model, let's say a GTR model for non-reversible data. But as we can see, the effect is not so much. The maximum average probability percentage error, it was in all cases lower than 5%. And on the right, we see the distributions. The same was for Bayesian and real-time. A little bit, the difference was a little bit higher in real-time. And as we can see on the distributions on the right, which is on this one, on letter C, figure C, we can see that the difference is not so much, it's close to 0. And on the right, D, figures D are the difference against the true times. We see that there is a little bit of dispersion, and it is a little bit higher for real time. Then we compare the branch lengths, not the times. We wanted to see if what was happening, if the branch lengths from the data sets were very different or not. These are maximum likelihood branch lengths compared. And as we can see, the R square was very similar, so the correlation was high in all cases. And we can see the dispersion is not so much. So this is on these 2 scatter plots is an example of just one data set, but on the right we have the average R square for the 150 data sets. For 150 datasets AR and 150 dataset independent rates. So we see that the correlation of branches is high. Then we wanted to check if that was the same, what we can see when we use the non-stationary data. As you can see on the scatter plots, using the GTR data to estimate non-stationary data, to estimate non-stationary data data sets produce a higher error than using time reversibility. So using non-stationary data, the GTR model for analyzing non-stationary data has a higher impact than time reversibility. As we can see in Bayesian, the average is 8% of error, and in real time it was around 10%, a little bit higher. And also, as we can see, the density on the right, on the most right figures, it's higher than the time reversibility when we were comparing the time reversibility against the true times. So we then again wanted to check what was, if the reason could be that the branch lengths were very different. And in that was the case. As we can see, the correlation is also very high, but the dispersion was higher than for time reversibility. So interestingly, and then we wanted to check what happened when we include calibrations to the analysis. So for that, we calibrated the nodes in which the substitution, in which we simulated a shift on substitution rates and base composition. And we found that using calibrations produces better estimates than without calibrations. So this is good because if we have good calibrations, we can even reduce further the impact of analyzing this type of data that violates the assumption using a GTR model. And the same in the case of of real time, sorry, the error was reduced around 5% for real time. And finally, we wanted to check the coverage probability of the confidence intervals.
Jose, excuse me, just
yep.
one more minute. Just.
All right. So Bayesian probability coverage was very high, was 96%, and real time was a little bit lower, 94%. We assume that this difference is because in Bayesian we use the proper priors to estimate times, and in real time we don't use priors. So that could be a difference. So to conclude, we show that violating the time reversibility assumption does not have high impact. In contrast, stationarity has higher. Fossils can help. And these conclusions are consistent with Tao et al., in which the substitution model has a low impact on time estimates. And the primary reason is the good performance of GTR plus gamma and the high linearity of branch lengths from the mismatch model. And that will be all. Thank you. I want to thank my lab and my collaborators and Temple University. And that will be all. Thank you.
Thank you, Jose. I don't see questions. We have still time for one small question. Nobody interesting. Everybody's hungry. So I'm not from the field, so some of the things you talked about are very weird for me. You talk about calibration. Could you just, in 2 seconds, tell me what that means?
Of course. When we analyze data to estimate divergence time, we need to put the relationship of times into absolute times. So for that, we use geological times or fossil calibration. So that will make the times into absolute times, because otherwise we're going to have only relative times without the absolute. So we use calibrations to produce absolute times.
So, but you have to basically, you are introducing extra information in the.
Yeah, exactly. You include extra information that you have a priori.
But that would be more general. So you may have more information or more, for instance, the actual mutation rates or something. You may have other biology information you may introduce.
Yeah, exactly. You may have your rates of substitutions and also some knowledge in a priori of the origin of the different lineages based on the fossil record. So this is another prior that you can add up into analysis.
Okay, thank you. So thank you, Jose. I think we should go on. Our next speaker is, if I can find it, yes, Jason Fan, I think is the right pronunciation, is going to talk about matrix factorization reloaded, flexible methods for inputting genetic interactions with cross species and site information. Jason, the floor is yours.
Great. Can you confirm that you can see the slides changing? Hello?
Your point.
Yep, you can hear me?
Yes.
Perfect. So good afternoon, everyone, and good morning to my friends in the US. My name is Jason Fan. I'm a third-year PhD student at the Center for Bioinformatics and Computational Biology at the University of Maryland, College Park. And today I'll be sharing with you our work on matrix factorization reloaded, flexible methods for impeding genetic interaction. This work was done in collaboration with a fellow student, Cindy, our collaborator, Mark Cravella at Boston University, and Max Leis is my advisor at UMD College Park. Allow me to quickly remind you of the main biological phenomena of study in our study, which is epistasis, or the genetic interaction. Genetic interaction between a pair of genes A and B occur when a double mutant is observed or measured to have an unexpected change in fitness when compared to single mutants. The strength of this interaction is measured by a real valued genetic interaction score that quantifies this genotype to phenotype relationship. And interest in modeling this relationship began first with classification in predicting extreme negative genetic interactions, or more specifically synthetic lethal interactions, in which a double mutant essentially it is unexpectedly essential. But more recent studies have tackled the more challenging problem of regression, in which both the sign and magnitude of these interaction scores are predicted. So this is the focus of our study. The computational side of this work is obviously complementary to huge in vitro methods in mapping out interactions in the lab, culminating, in I would say a decade-long effort in baker's yeast, in which 90% of the non-essential genome has been mapped, yielding an unbiased map of the cell. But there are ongoing efforts to measure genetic interactions with respect to phenotypes beyond simply fitness. So, for example, groups have started to study how these double mutants have unexpected expression, or even measure genetic interactions between more than 2 genes, so say triples of genes. Orthogonally, there have also been developments in protocols to measure GIs in other model organisms, so say across different yeast species, and also across different contexts, so say the cancer cell lines for potential translational applications. The most successful computational efforts have been in baker's yeast, as I mentioned earlier, where data is very abundant and have utilized very rich sources of side information, specifically the gene ontology, to predict genetic interactions. So I'll give where credit is where credit is due. In 2018, Model developed D-Cell, an interpretable neural network that predicts genetic interactions in baker's yeast with very high accuracy. Though successful, there remains several challenges that if we wish to predict genetic interactions in the future. First, gene ontology labels are not equally abundant across different model organisms. And 2, it's unclear how these prior approaches, which have utilized this very rich resource, will perform in other species or new phenotypes where the data, both labels and features, are very scarce. So our study set out to tackle 2 questions. And first is, how can we model genetic interactions without relying on specific side information? And 2, how can we exploit GIs in data-rich species, for example, in baker's yeast, to understand GIs in new species, say different yeast species, or say in cancer cell lines? To that end, we introduce extensible matrix factorization, a flexible matrix factorization framework for imputing genetic interactions, EMF, which we call, can flexibly and optionally exploit cross-species information and arbitrary side information to better impute interactions. In our experiments, EMF outperforms the state-of-the-art deep learning model and is much faster to train. Moreover, by a careful ablation analysis, we find that exploiting cross-species and side information improves GI imputation where data is scarce. So these are settings that are likely to occur for new experimental protocols. Before I dive deeper, let me remind everyone some details about matrix factorization. So the prediction are from now on, I'll say the imputation problem arises from a partially observed gene-by-gene matrix of genetic interaction scores X. Matrix factorization assumes, if fully known, X would be effectively full rank. So therefore, we can recover X, the missing entries of X, by a low rank factorization with respect to the observed values. And to predict the missing value, you simply take the inner product of the learned latent factors. The REMF framework extends this basic matrix factorization approach in 2 ways. First is we can exploit cross-species information, or more specifically, genetic interaction datasets across related model organisms to better impute GIs in a specific target species. And 2 is we decouple the reliance from Go, and our framework allows the exploitation of arbitrary side information for example, PPIs from high throughput experiments that require less researcher input, supervision. Oh, okay, that's okay. So the EMF framework, or cross-species matrix vector, has a core component, which is cross-species matrix vectorization, and it is set up as such. The motivation is that in a new target species, genetic interaction data from new experimental systems is likely scarce, and in a related source species, for example, say baker's yeast, data is very abundant. And our goal here is to automatically exploit the shared structures and similarities between the source and target species to better impute missing values in the target. And the key idea here is that we want to jointly factorize the target and source matrix to better impute the missing values. This is not trivial because there is no one-to-one correspondence between the rows and columns between the two matrices, since the genes in different species are different. And our formulation specifically involves linking the latent factors with any reasonable similarity measure between target and source genes. So, for example, BLAST would be a canonical similarity measure. And this formulation basically amounts to trading off reconstruction of the target and source input matrices with a regularizer that links the latent factors. So this is the first component of our framework. Our framework consists of two other components. So the first is EMF allows for the modeling of explicit latent per gene bias terms. And two, EMF allows for the incorporation of arbitrary site information. And by arbitrary, I mean that the site information that can be encoded via kernels. So these have, for example, PPI kernels have been used to study protein function. These components are all differentiable and therefore can be freely and flexibly composed, yielding a family of EMF models. And in essence, EMF unifies several existing matrix factorization models and extends them into the cross-species setting. Surprisingly, even a simple EMF model outperforms the state-of-the-art deep learning model and is much, much faster to train. We compared EMF to gene ontology-based methods, so D-cell and this other method, ontotype, on a data set of 3.3 million genetic interactions in baker's yeast. In regression, EMF models has a 15% improvement over D-cell and Pearson row. And in classification of extreme negative genetic interactions, EMF has a 16.3 improvement over D-cell in terms of AUPR. EMF takes less than a minute to train, whereas D-cell takes 2 to 3 days to train on a different data set of 7 million genetic interactions. So you might be asking the question now, how does this matrix factorization algorithm outperform a neural net? And I want to make a very key point here, is that we're not making the claim that EMF is more expressive than D-cell, but EMF is fundamentally different and much faster to train. Therefore, automatic hyperparameter selection, which has been an issue in training, is always an issue for grad students training these models, is feasible, but it's also easy. This means that we can do this and profit and trade off training time with selecting better hyperparameters in an intelligent manner and get the most out of both our data and the model class. So we view this automatic hyperparameter optimization step necessary and optimal for optimal model performance. Moreover, it's a very useful tool in comparing a family of models, especially if we want to get an apples-to-apples comparison. So with this tool in hand, we performed a careful ablation analysis of all these model components to kind of understand how cross-species information, site information improves GI imputation across different kind of settings. To do this, we ablate these extensive EMF matrix factorization, EMF components, specifically kernelization of site information, cross-species information, and also biases, and vary the amount of training data to simulate data settings that we would likely see in new experimental protocols. We perform our data set on a matched pair of genetic interaction data sets in baker's and fish and yeast. So these are chromosome biology genes, gene data sets. And our results show that EMF components improves model performance. So on the right, I'm showing you a plot of a fully fledged EMF model in which all three components are used. And we see that the improvement is the greatest when data is scarce, so specifically when only 10% of available genetic interactions are observed during training. I would like to refer you to the manuscript that will be coming out to see the details and kind of the lattice of different models that we analyzed. So to conclude, we introduced extensible matrix factorization, a flexible framework that does not depend on specific site information. It is very fast to train and outperforms the state-of-the-art neural network. And it enables the optional and flexible exploitation of cross-species and site information, which we find in our experiments to be fruitful, if not necessary, for new experimental settings where data is scarce. For future work, we would like to combine multiple sources of site information, maybe optimize and gain more performance by using multiple kernels. So in our work, we use PPI kernels. And two is like, we would like to kind of take this factorization approach to explore how we can impute interactions between more than a pair of genes. We view this to be potentially very fruitful because it's currently still experimentally feasible to explore the combinatorially many interactions between n tuples of genes. And then I want to highlight that I was really interested in Elliot's earlier presentation, and certainly maybe some ideas about incorporating phylogeny into the optimization would be very interesting. So I'd like to take this opportunity to thank my collaborators at College Park and Boston University, as well as the center where I do our work, as well as our various funding agencies. Please ask me any questions you have, and I'm having some trouble with the portal, so tweet at me or send me an email. I'll be available for the rest of the day. Great, thank you.
Thank you, Jason. So we'll still have the time for a couple of questions, but as lately we don't have questions in the chat. Again, this is far from my understanding, but I have perhaps a more general one. You was using sequence comparison. You cited protein-protein interaction networks that are very popular. Which is the kind of information that you can use a part of these things? Gene ontology, for instance, is there, but any other kind of network or disease, for instance, relationships or something like that?
Yeah, so our formulation is based on one, being able to transfer knowledge across different species in which it's difficult to get a correspondence between genes. So any similarity measured between genes, they will work. And then to make the model even better, you simply need notions of similarity within a species. So that can be anything that is arbitrary. So for example, PPI networks is useful. You can take like co-expression networks or even signaling networks. There's some things that we didn't have time to explore, but in future work, we'd like to kind of figure out how do you combine these different sources of site information to get the best out of the model. Okay,
no, that makes sense. Did you try other further species, not just two yeasts?
Yeah, so the current restriction with respect to these gene-gene interaction networks is that the data is very sparse for these other species. So kind of our goal here was like with this small species, we can show a proof of concept in which we can link the factorization via a pretty I would say simple linking scheme. So in the future, we would love to try some linking species that are more not as related.
Okay. Thank you, Jason. I think we should go on. Our next speaker, if I can find it, is Kerem Ayöz. I don't know the right pronunciation for the Bilkent University Ankara. Is that?
Yeah.
No. Sorry, is that the right one? And the talk is about a completely change of issue, the effect of kinship in the re-identification attacks against genomic data sharing beacons. Kerem, go ahead.
Okay, hello everyone. I am Kerem Ayöz. I am a master student in the Bilkent University, and I will present our joint work with Miray Ayşen, Erman Ayda, and Ercüment Çiçek. And in 2016, Global Alliance for Genomics and Health introduced the Beacon project. Beacons are the database of human genomes, which provides a secure and systematic way of sharing genomic data. In this system, users can send queries about particular alleles and receive binary response for their queries. For example, user can ask a question like, is there a participant carrying nucleotide C at the 1000th position of chromosome 1? And system responds with either yes or no. Here, yes means that at least one of the participants have that queried allele, or no means that none of the participants have that queried allele. And several studies show that this system is vulnerable to re-identification attacks, which I will describe soon. And in this type of attacks, attacker aims to obtain the membership information of a particular victim. And some studies show several countermeasures to overcome these vulnerabilities. For example, constructing larger beacons or sharing some portion of the beacon, or not publishing any metadata about beacon, or having uniform ancestry, or randomly flipping the responses, which means that giving wrong answers to users sometimes. But the main problem is these countermeasures decrease the utility of the beacons project. And in this study, we analyzed the effect of kinship relationships among beacon participants against re-identification attacks, and how this might be an effective and harmless countermeasure in terms of the utility of this project. And let's start with describing what's a re-identification attack. As I said, re-identification attacks aim to obtain membership information of a victim. First one was proposed in 2015 and it's called SP attack. It is based on likelihood ratio test, and here we have two hypotheses. The H0 is a null hypothesis in which victim is not in the beacon, and H1 is the alternate hypothesis in which victim is in the beacon. In the likelihood of null hypothesis, attacker asks queries from 1 to n, and xi here is the received response from the beacon. It's a binary variable. This means that attacker obtains yes answer, and which means that at least one of the participants have the queried allele. So logarithm of 1 minus dn, where dn is the probability that none of the n individuals has corresponding allele, goes into the sum. And if xi is 0, which means that none of the participants have that queried alleles, so logarithm of dn goes into the sum. And alternate hypothesis is similar to the null hypothesis, but we have two small differences. First, we know that in the alternate hypothesis, victims in the beacon, so dn changes with the dn minus 1, where dn minus 1 is the probability that none of the n individuals except for the victim having corresponding allele. And here delta is the sequencing error for the queried allele. And after calculating likelihoods, the lambda value is calculated from these likelihoods, and null hypothesis is rejected if this lambda value is less than some threshold t. And since our queried SNPs are IID, this threshold t can be found theoretically. And from these threshold values and lambda values, the power value is calculated to measure the power of the attacker. And more advanced algorithms have been proposed after SP attack, and optimal attack is one of them. It was proposed in 2017. Additionally, it uses minor allele frequency information from a population. In optimal attack, some of the alleles might be hidden from the attacker, so attacker starts from the non-hidden allele, which has the lowest minor allele frequency value, and continues in the ascending MAF order. Here, since our SNPs are not IID anymore, we are starting from the rarest SNP. Power calculation is based on the empirical distribution of the data. And only difference in the lambda calculation is that the DN values are specific to the minor allele frequency of the queried allele. And in the optimal attack, much less queries are needed compared to the SP attack. Even 5 or 10 queries are enough to identify the existence of a victim in a beacon. And even better algorithm, query inference attack, was proposed in 2018. Additional to MAF information, correlations between SNPs are also used. And these correlations are based on the LD values. Pattern is similar. Attacker starts from the non-hidden allele and continues in the ascending MAF order. But this time, by querying a single allele, attacker can obtain information about multiple alleles, not just single one. And in the lambda calculation, as you can see, we have this same part with the optimal attack, and additionally, we have the information came from the neighboring SNPs to the queried SNP. and it is one of the best algorithms in terms of number of linked queries. Even 3 or 5 queries are enough to determine the existence of the victim. And we used optimal attack and query inference attack in our experiments, and we have used 2 different data sets. First one is C population of the HapMap project. It contains 41 different mother, father, and child trials, but not any grandparents. To simulate the effect of having grandparents, we scientifically generated the families, and we used an algorithm which is haploblock array based on the MF information. And let's describe how our algorithm looks. Our algorithm consists of 5 parts. In the first step, child sublobe is taken and divided into 2 strands. And in the second step, the heterozygous alleles are shuffled. And in the third step, these strands are distributed mothers and fathers genomes equally. Then to sample the remaining strands, we use the MF information and sample by the allele frequencies and obtain mothers and fathers genomes as a result. And to generate the grandparents, we use the synthetically generated mothers and fathers genome and input them to our algorithm. So we have obtained the grandparents genomes with that method. And we carried out 4 different type of experiments. In the first one, we have no parents of the victim in the beacon. And in the second one of the parents of the victim is in the beacon. And in the third one, both of the parents of the victim are in the beacon. And in the fourth one, grandparents of the victim are in the beacon, but we use different combinations of the grandparents. And let's move on to our results. As I said, the attack's power measured with a power value. Here you can see the power plot. In the x-axis, we have number of queries, and in the y-axis, we have the power values. And for example, we can identify the 60% of the beacon members correctly, and power equals 1 means that attacker can identify 100% of the beacon participants. And when we add mother or father to the beacon, additional to the victim, we see that power shifts to the right, which means that attacker needs much more queries to identify the existence of the victim. But when we add both of the parents to the beacon, we saw that these identification attacks failed, and in some cases power never reaches to the 1 and stays very low. And this result is expected since a child's minor allele either come from mother or father. When we add mother and father both to the beacon, attacker cannot distinguish between father, mother, or the victim. And in the synthetically generated dataset, we obtain a similar trend, but even more optimistic. Power shifted to the right with addition of single family member, and addition of 2 family members caused re-identification attack to fail. And even in the QI attack, addition of 1 family member damaged the attack substantially. And finally, we measured the effect of having grandparents. In the first row, you can see the effect of having single grandparent. First one represents the having victim's mother's father, and second one represents the victim's mother's mother. And having single grandparents to attack the victim, but in the second row, you can see that having 2 grandparents effect of having 2 grandparents in the beacon, and these plots are similar to having only mothers in the beacon. And however, they are also even worse because having both of the grandparents in the beacon bring diversity to the beacon, and even damage the identification attack more than the having only mother or having only father. And we also measured how adding additional family members affects the utility of the beacon. And here we defined utility loss as the fraction of additional yes responses that arise due to the addition of family members. Here, yes responses are important for us since they carry the information for the attacker. And in the table, you can see that we have 2 different groups. The control group are the people who are not in the beacon, and case group are the people who are in the beacon. And as expected, control group have higher utility loss since they are not in the beacon, and we obtain 7.96% utility loss in the worst case, and this is an acceptable result. And when we compare with the other countermeasures like random flipping to responses, which is we obtain a better and lower utility loss in our experiments. And we also quantified the utility loss of the beacon responses considering the alleles with the low MAF values. There's an inverse relationship between variance disease odds ratio and its frequency, which means that the SNPs with the lower MAF values are much more important when deciding these diseases. And when we look at our plot, we have the MAF threshold on the x-axis and fraction of additional yes responses on the y-axis, and we can see that when MAF threshold gets lower, we have a much lower utility loss, which means that our proposed technique does not affect the rare and important SNPs that much, and we have even smaller utility loss in these SNPs. And currently it's shown we saw that beacons are vulnerable to re-identification attacks, and also there are newly proposed attacks like genome reconstruction attacks. We have a preprint about that. You can see it on the archive and welcome to read. And in the future work, we might consider to see the effect of having family members against these genome reconstruction attacks. And the proposed solutions to overcome these vulnerabilities decrease the utility of the beacons project substantially. And with our proposed technique, effects of re-identification attacks are mitigated, and even these attacks are failed. We also think is that we have medical protection for both victim and the additional relatives. When we add mother to the beacon additionally, not only child is protected, but also mother is protected or father is protected since they share common alleles, and attacker cannot distinguish between them. And as I showed, the utility of the beacon is not affected substantially compared to the other methods. We have an acceptable utility loss in our experiments. And thanks for listening. I am happy to answer your questions. You can reach me via my email. And thanks to our supporters for supporting us.
Thank you, Kerem. I understand that it's too late. There's no questions on the chat either. And we're almost finishing. I have perhaps a devil's advocate question. If I change my strategy for attack, instead of finding a single person, I try to find a family, you're lost.
Sorry?
If my strategy of attacking is instead of a single individual, trying to find a family, then
this might, yes.
you have a problem.
No, because family members have common alleles, we might attacker can change his strategy.
I mean, there is many data sets, and you have trios and all these families already there. Mm-hmm. So it's not negligible to perhaps a different strategy, because basically you are confusing the attacker because you have similar sequences. But if you find a group of that,
but not always all the family members send their genomes to the beacons. This is our proposed technique, but I don't think attackers can change his strategy because the family members share alleles, and cannot distinguish between them.
Yeah, it depends on how you want to attack. So thank you. No more questions from my side, but I have more, but I think we are just in the time for closing. Let me thank all the speakers from this session, the previous one, for the interesting talks, the interesting work, and thank you everybody for coming, and all the participants as well. Thank you, and bye.